Hi, everyone. Thank you so much for joining us today. And thank you to LabRoots and Bio-Rad for giving the opportunity to talk to you about our experience with Droplet Digital PCR at Biodesix. So the title of my talk today is Blood-Based Detection of EGFR and KRAS Mutations for NSCLC patients using multiplexed Droplet Digital PCR. My name is Hestia Mellert. I am the Senior Director of Development at Biodesix. I've been with the company for just over 5 years now and have had a major part in the development of many of our tests, including Droplet Digital PCR applications. We'll start out with a brief overview of Biodesix and what we do as a company. Then we'll move into the laboratory workflows for ddPCR applications, and we'll focus our attention on a laboratory-developed test called GeneStrat that looks at somatic variants in DNA as well as RNA fusions. Then I will introduce the next generation of Droplet Digital PCR, that being the QX1 instrumentation. I will tell you a little bit about our experience early on with Bio-Rad as they were developing this instrument, as well as now in the present as we have these instruments in our laboratory, in our clinical laboratory setting. And finally, we'll close out the session talking about a few case studies using Droplet Digital PCR in the clinic, specifically looking at how this technology can be used both to diagnose treatment, but also in monitoring treatment outcomes. Biodesix really has two core business units, and they're represented here on this slide by these two circles. We have a commercial side as well as a pharma side. Both of these are supported by our two clinical laboratories. The first one's located in Boulder, Colorado, in our corporate location. And this laboratory has certifications with CLIA, CLEP, and is ISO 1345 accredited. And then we have an additional clinical laboratory doing testing out of DeSoto, Kansas as well. On the commercial side of things, so we're looking at the white circle now in the Venn diagram, we've got 6 now on-market tests, actually. We've just brought up our SARS-CoV-2 total antibody test, and that complements the molecular testing that we are doing with Droplet Digital PCR. And in addition to that, we have 2 suites of tests. One looks for to help with the diagnosis of indeterminate nodules in the lung. Those are the Notify CDT and XL2 tests. And the second is following diagnosis of cancer, helping to understand the molecular underpinnings of that cancer using liquid biopsy, which is the GeneStrat test, which we'll go into in more detail in this presentation, and the VeriStrat test, which complements this test in prognosticating. So in addition to those tests that are available to physicians as on-market laboratory-developed testing, we also have a robust pharma partnering business, which utilizes our clinical laboratories. We do testing anything from early preclinical studies all the way through phase 3 trial sample testing using these technologies. These are the core platforms that we use in the laboratories. We consider ourselves generally platform agnostic. That is, we bring platforms and technology into Biodesix because it answers a key clinical question in the lung care continuum. And so because of that, you can see here that we have technologies both supporting genomics and proteomics, and really do span the gamut from targeted PCR, like Droplet Digital PCR, to genome-wide NGS platforms. We look at proteomics with MALDI-TOF mass spec, LCMS, as well as ELISA testing. And if you dig deeper into what those technologies really do provide to the community, it's a nice complement of both looking at the tumor itself and profiling the molecular underpinnings of tumor biology, as well as supporting that with the immune response or the body's response to that tumor immune profiling. And so these technologies all together can really give us a lot of different types of tools to answer those burning questions. This is a map I'm showing you specifically for GeneStrat, where our laboratory developed tests are used across the United States. And you can see that although we do have testing happening across the entire United States, we tend to focus and have a large number of tests coming from that northeast corridor there, where there is a high prevalence of lung cancer in our communities. So let's focus now a little bit more on our genomics, and specifically our Droplet digital PCR test. That's the GeneStrat test. The GeneStrat test addresses through liquid biopsy mutations that are common and actionable in non-small cell lung cancer. And you can see those mutations here on the slide. We look for EGFR sensitizing mutations, exon 19 deletions, L858R. We look for the more uncommon mutations in EGFR in exon 18 and 20, as well as T790M resistance mutations. And in addition, we can look at mutations in KRAS and in BRAF. Those all happen by looking at the DNA, the circulating DNA or cell-free DNA. And we do this by supplying our test sites with the Streck cfDNA2, which is a blood collection tube that's specifically designed to stabilize nucleic acid or cfDNA for ambient temperature shipment. And in addition to looking at mutations from DNA and small insertions, deletions, we also utilize the Streck RNA tube to look for circulating RNA as well. And this allows us to look at those actionable mutations that are actually due to rearrangements in the genome and generate fusion transcripts. So there, we're looking for ALK fusions, RET fusions, and ROS1 fusions. And you can see the partners that are specifically measured underneath each in the table provided. So the pre-analytic pieces of GeneStrat include a test going out to the community or the physician that is interested in ordering the test. They draw these blood tubes for us, and then they can ship them back to us at ambient temperature for testing in the centralized laboratory. Once those tubes arrive to the laboratory, we initiate the GeneStrat workflow, which I'm describing here to you. The first thing that happens is we accession those samples into a laboratory information management system and give them a unique de-identified number. And then that is carried with them throughout the process. The first step is extracting the nucleic acid. So we have extraction methods for both RNA as well as DNA. And once we have that nucleic acid, the RNA is converted to complementary DNA or cDNA by an RT step. And then that's where the Droplet digital PCR workflow is initiated for the GeneStrat test. And up till very recently, we were using the QX200 system. That is a relatively manual system. We have the manual generator of droplets there. Then we go into a thermal cycling step on a standard thermocycler. And then we read those droplets following thermal cycling. And then we are able to now analyze each individual droplet using two-dimensional plots like I'm showing there. That process can then be essentially analyzed and reviewed, generating results for GeneStrat. So either identifying that there was a mutation of interest, for example, an EGFR mutation in that circulating cell-free DNA sample, or that there was no mutation detected. And the whole process is designed to work really within 48 hours. And I will show you on the next slide what that looks like in the lab in practice. turnaround time of GeneStrat is displayed here in this pie chart. You can see in the dark blue that about 50% of the samples that come into the lab upon arrival can be then reported back to the physician and the patient within 24 hours, and an additional 40% by 48. And so this really provides us with fast turnaround times that allow the physician and patient to make informed decisions with molecular information about their tumor upon first treatment. And this has been a really kind of important characteristic of being able to use this droplet digital PCR technology in our workflow in the lab, is the turnaround time that physicians are really looking for to make informed decisions about treatment. To focus a little bit on EGFR, so this is our ddPCR testing specifically for EGFR. You can see here the clinical sensitivity, specificity, and overall concordance for each one of those actionable mutations that I mentioned earlier, as well as our C797S resistance mutation. And what you can see here is that the clinical specificity in our initial studies for validation is extremely good, really demonstrating that the technology doesn't sacrifice specificity, although it does absolutely have wonderful sensitivity levels that are really required to have application in liquid biopsy, where the cell-free components from the tumor can be at really quite low levels in circulation. And so the droplet digital PCR is really a good tool for liquid biopsy testing because of its great specificity as well as superb sensitivity. This is an additional study that we did once we were on market with a partner. These were the AURA3 samples from AstraZeneca. And so you can see in real world settings as well with the AURA3 trial samples, our specificity was very near 100% in each case. And our sensitivity was also quite good for the different EGFR mutations. It's interesting to note here that we identified, along with AstraZeneca, that there are certain patients, I should say, that do not shed cell-free nucleic acid. And what we identified was that independent of which type of technology you might use on those patients, they're considered non-shedders. And although their tumor may contain some EGFR mutation, that liquid biopsy is unable to detect it. Okay, so now let's move into the QX1. As I mentioned earlier, this is the next generation from Bio-Rad for digital PCR. And I wanted to introduce you what is new about this technology. There's quite a couple of exciting developments. And then I'll give you the experience that we've had both during the development and now with the instrumentation. So I introduced this a little bit earlier. This is the QX200 system with a manual droplet generator. And as implemented in our laboratory to run the GeneStrat liquid biopsy test, it takes about 30 minutes of hands-on time to generate droplets. Then you take those droplets in their 96-well plate and put them into a thermocycler, and that cycling program takes about 2 hours. That would be hands-off time. And then a technician would need to come back to the laboratory and transfer that plate onto the reader, which is another about 2 hours worth of time. So if you're crunching the math here, it takes about 12 hours of tech time. Some is continuous when they're doing the droplet generation, and some is transferring plates in between these different steps to do 5 plates in a day. And so although this has been worked into our workflow in this manual way in the clinical laboratory, of course, it's not always ideal to have so much interaction with the workflow. And the more we can automate things, the better it is for our clinical applications. And that's where the QX1 really does shine. So here, what this figure is showing you is that the generation, the thermocycling, and the reading are now all in one automated workflow within this larger box called the QX1. So the QX1 also has the capability of taking up to 5 plates at a time. And so you can load your 5 plates and essentially walk away from the instrument. And the additional benefit to the QX1, above and beyond the throughput benefits that you get in the automation, they've added additional colors. So the QX200 manual system had both hex and FAM channels. And now we have 4 different channels that are able to be used within the QX1. And so you can design assays that are more complex and get more information out of a given run and a given amount of sample. And so that's another great benefit to the new technology. The QX1 ddPCR system expands upon the most proven digital PCR technology for unmatched precision and sensitivity in absolute quantification, with automated and walk-away operations for multiple plates, and the system's easy to use, supports regulatory compliance, and enables more answers per sample with advanced multiplexing. So you can see here with the reagents now as well, and there are really easy to use oils that can be imported into the instrument with barcodes as well as barcoding on the plates. So you can really have traceability of your specimens and your reagents throughout the process. So all in all, we've taken essentially a workflow where if you wanted to look, for example, 20 targets, because there's 2 channels that would require 10 plates. And if you have 2 systems running in parallel, for example, you could get that done in about 12 hours of tech time. And now we've taken that same thing, and with a capacity of 5 plates, being able to input into the system and walk away, and 4 targets, you've greatly reduced that time to about 2 hours of actual hands-on tech time. So you can see really the benefit in throughput and the interaction with the instrument have improved with the automated system. And the other benefit, of course, to automating is that you can get more consistency in your workflow. So droplet generation can be manually a more difficult process that requires trained technicians. We are lucky at Biodesix to have a staff of very talented technicians that can generate droplets manually very efficiently. But the nice thing about having a system like this is that you don't have to worry about that piece. You take your master mix, you mix it with your template of interest, and the instrument does the rest. So you can load it onto a cartridge that looks much like a fancy 96-well plate, essentially scan the plate into the instrument, again up to 5, and that's it. The instrument does the rest until data is ready for you to analyze. So just to review some of the key benefits that we've found while we started to use this instrumentation, it's an automated walk-away operation. It has a 5-plate capacity. It has advanced multiplexing capabilities using 4 colors now, rather than the 2 that were on the original QX1 systems. And just to kind of elaborate on that a little bit, that doesn't mean that you can only look at 4 different targets. In fact, there's a lot of different ways that you can mix probes and change their concentrations, essentially, to get even more information. So increasing from 2 colors to 4 colors can actually be quite a significant increase in the amount of information that you can gain from your sample. So those 2 types of things could be amplitude multiplexing or probe mixing, for example, as is presented there. It's easy to use, of course, as I mentioned earlier. The less time you need to take training technicians to make sure that you've got quality data, of course, that's definitely a benefit. And then it also has CFR 21 Part 11 compliance for its software, and the barcodes help you to track consumables and allow you to manage your samples as they go through the workflow. Okay, next we're going to move into a few slides that I put together that presents some of the data that Biodesix generated alongside Bio-Rad during alpha and beta testing of the QX1 instruments. So initiated with alpha testing, where we focused on some of the mutation tests that were important, of course, to us looking at non-small cell lung cancer. And so we decided to start out by looking at our EGFR and KRAS mutation tests. And it's important to note here that in the past, our GeneStrat test is using individual assays to look at the mutation in one channel and then the wild type control in the other. And so in that way, you have to run additional assays to look, for example, at each variant of interest, KRAS G12C, different than KRAS G12V, et cetera. And so the nice thing here was that we were able to use the 4 color channels to actually multiplex now to more extent and be able to identify different mutations within the same liquid biopsy sample and assay run. We found that the assay setup was relatively straightforward. Again, mixing your assay of interest with your template and then putting it into the instrument. It doesn't get much easier than that. And the software, we were able to work with them on the software that they have developed, and they've really made it a nice interactive environment for us to do the data analysis on the back end. Once the alpha testing was done, which happened at Bio-Rad in Pleasanton, California, our feedback and feedback from other laboratories was taken, and they made some adjustments. And then they contacted us again when they were ready to place some instruments for beta testing. And so these pictures here are actually our technical director and some of our lab technicians and research associates being trained at, on the beta instrument at Biodesix in Boulder, Colorado. And during the beta test, we ran approximately 21 plates, or over 2,000 different targets through the instrument over about a 3-week period of time back in July of last year. So what we found, again, was that the instrument was easy to use. It was easy to train individuals to use the instrument. We really appreciated the fact that it conserves sample because you can get that additional information out of the same aliquot of sample. The data analysis was pretty straightforward, and we liked the fact that it still allows you to look at the 2-dimensional plots that we're used to looking at with the QX200 system, as well as getting additional options to look in more 3-dimensional space now that there's 4 targets per reaction, or 4 channels, I should say, per reaction. This slide shows some data that we've generated while we were in beta. So this was an assay that was generated with EGFR liquid biopsy testing in mind. It contains a wild type EGF probe that is in the region of C797S. The assay also contains probes that detect T790M, L858R, and C797S, and there's actually 2 different nucleotide changes that are commonly causing C797S, so you can identify both of them, as you can see there. So this was one of the first examples of a higher multiplex test that employed all 4 channels of the QX1. And you can see on the top, that's how you can look in 2-dimensional space for the droplets that are negative and positive for each of the targets of interest. And then on the bottom is another view of the same data. This allows you to look at the concentration in copies per microliter of each one of your channels or your mutations of interest, and calculate the concentration within your sample. This assay was also done with the 4X supermix from Bio-Rad. This is another improvement that isn't necessarily directly related to the QX1, but it is a really nice improvement because it allows you to use more sample as well. And this is becoming more and more obvious as we look at liquid biopsy samples, that the more sample that you can put into the system, the better your sensitivity or your chances of being able to see that rare mutation will be. So by increasing to a 4X master mix, that allows you to put more of the sample into each reaction. During beta, we were also asked to run some experiments for Bio-Rad that specifically looked at the accuracy of the test. So here we are using the National Measurement Institute standard on the QX1. And you can see that our average concentration was right at around 2,000 copies per microliter. 2,100 was the target concentration that was advertised by National Measurement Institute standard to be the absolute concentration of the sample. And then in addition, across these specimens, we saw a CV of 1.6%. So you can see here really a lot of nice consistency and precision across multiple reactions using this standard. Next, we tested some of our own specimens that we have in our laboratory. We wanted to see how the instrument would perform when we used actual clinical samples. And so here are a few different examples. Our duplex test is in blue, and then the quadruplex tests are in red and green here. And what you can see is that running the QX200 in a duplex type of test for each one of these variants is very equivalent to what we were able to obtain running the same sample through the QX1 workflow. And that was true both for looking at positive samples that were detected using all of those different systems, as well as the different multiplexing strategies on the QX1. But it's also important to note that we didn't see any false positive or any signal coming up in the QX1 versus QX200 in these samples. This is additional information in addition to the EGFR assay. We also developed an assay for KRAS with Bio-Rad, and this assay looked at C, D, and B, G12C, D, and B. And so here again, these are clinical specimens. And what you can see here is that there's really nice consistency across both positive and negative detection of KRAS mutations in NSCLC patient samples. So again, just demonstrating that the QX1 system functions much like our current approved GeneStrat test that's run on the QX200 on clinical samples. Okay, so the last piece of the talk, we're going to dive in a little bit to some data that uses Droplet digital PCR out of liquid biopsy or blood samples for the purpose of monitoring. The first is a case study that was published in 2019 with Dr. Bowling. Here we're looking at a patient that is a female never smoker that presented with new onset seizures and was diagnosed with extensive brain and bony metastatic positive lung adenocarcinoma. And upon molecular testing with GeneStrat, initially she was found to have an EGFR exon 19 deletion mutation. She was initiated on treatment at that time. The treatment was generally a first generation tyrosine kinase inhibitor. Of course, now it may be osimertinib. So she was given that treatment and then acquired resistance in the EGFR T790M mutation as it emerged. At that time, she was switched to osimertinib, and over time then began to have progressive disease. And so we did look at Biodesix retrospectively at this specimen after she had discontinued therapy due to resistance and disease progression. For the resistance mutation, C797S, which we do now have developed in our CLIA lab and is a New York State approved assay for us that we're using in the trial setting. About 10% of NSCLC cases are C797S resistance mutations. Another common alteration upon resistance is MET amplification. So let's look more closely at what this looks like over time as we monitor these samples, looking at the cell-free DNA by droplet digital PCR. So at first you can see at time 0, she had, this was, our time 0 is when she started osimertinib treatment. So you can see that both the del19 mutation is high, the y-axis representing the allele frequency in the biopsy sample, and T790M is also present. And upon osimertinib, you can see at about just under 50 days, both EGFR signals have gone below the limit of detection of the test, or down to 0, if you will. And that remains to be so for about 100 days. And then upon further testing, we could start to see the recurrence of the disease, even from the liquid biopsy sample itself. And we saw both the EGFR, del19, and T790M come back, as well as starting to see the emergence now of a C797S mutation. And you can see that they really do increase pretty dramatically over time once the resistance occurs. And you can see the recurrence of disease as well in the associated scans. So this demonstrates in one case study how using a sensitive technology that has a relatively fast turnaround time with a non-invasive method like a liquid biopsy can help us to monitor the mutations that we're seeing from blood as these patients are treated and become resistant to disease, to therapy. This is another example. This is actually data that was presented from AstraZeneca. And this was part of their FLORA sample set, which is the first line for Tagrisso. And what you can see here as well are 2 examples of the dynamics of EGFR over time, looking at both the mutations del19 L858R as well as C797S and T790M. And you can see the dynamics will be different from patient to patient, but overall, the recurrence of the EGFR mutations from the cell-free DNA correlates with resistance to the drug and recurrence of disease. The one interesting thing I would say, though, about this study was that it also recognized that in many cases, ctDNA progression preceded or co-occurred with the resist progression of disease, or the PD. So that was in 66% of the patients in this study. And in general, the median ctDNA progression lead time was about 2.7 months on the progressive disease based on resist. And so it's interesting to note here that perhaps in some cases, the molecular signature in the blood can precede being able to see the actual progression of disease. So in conclusion, blood-based approaches have become an integral part of biomarker strategies in drug discovery and development. Of course, both droplet digital PCR as well as other technologies that must have really high sensitivity to be able to look for the rare mutation events that will be in circulation. And so this is really a key highlight of droplet digital PCR and its capabilities as it applies to liquid biopsy. Of course, it's not only whether or not the test is sensitive that is important, but also whether or not the results can be back with the physician and patient to be informative for next steps in their treatment plans. And that's where the turnaround time becomes really quite important. And we have, even with the manual workflow, been able to really implement a fast turnaround time for our physicians to this end. However, with the QX1, you can see from what we've presented and seen in here that the QX1 is going to be able to do that even better, right? So we'll be able to run 5 plates, for example, with a walk-away strategy that allows us to run more and gain information on more samples than what our current workflow would allow. The additional benefits of having a 4-target approach rather than the 2-target approach obviously also will increase the amount of information that one can get. And I'm really looking forward to seeing how the scientific community leverages those extra channels with unique types of assay design to really gain as much information as we can out of these limited sample types, like a liquid biopsy sample. We saw that the interplate precision was quite excellent with a CV of 1.6%. We saw that the copy numbers between the quadruplexed and the equivalent duplexed assay that we have approved were very equivalent to each other. And we also definitely appreciated the hands-on time being diminished by using this QX1 system. And since then, we have actually implemented the QX1s into the Biodesix laboratory. We have 2 new instruments that we are now working with. And really, as the assay developer at Biodesix, I'm looking forward to getting some time to work on those instruments and learning as much as I can about how they can help increase information content that we can get out of our specimens. So with that, I think I will close out with an acknowledgement slide. I wanted to thank, of course, the Biodesix team. Here we are being socially distanced with our masks as the laboratory team is still working around the clock now, not only to support our patients with NSCLC and with indeterminate nodules, but also to support those patients that are questioning their COVID status. And so this team here in Boulder is running our COVID ddPCR test as well as GeneStrat. And also wanted to say thank you to the Bio-Rad team from the digital biology group, who obviously have been a major support in us implementing Droplet digital PCR into the clinical laboratory. So with that, I think I will close out the session. And I am really looking forward to hearing if anyone has any questions or follow-ups for me. Thank you.